Alright everyone, welcome back, and we're continuing our discussion on Mendelian genetics, and today we're going to look at a couple more of Mendel's hypotheses for genetic inheritance. And we're going to start with the fact that for every inherited character that we have, every trait that we have, hair color, eye color, height, the organism has at least two genes, two alleles that control that character, and they get those alleles, one from each parent. So for every gene, that locus, that location on a chromosome is actually represented twice, which should make sense. We've got homologous chromosomes that carry the same information, and those homologous chromosomes, we get that double set, one from mom, the other one from dad. So when we look at your genotype, we've got to look at it as representing the genes that are present in the organism as a result, the genes that were originally passed from the mother plus the gene that was passed from dad. So when we write these genotypes conventionally, it's always two genes, so it's always two letters. All right, one of those letters or one of those genes comes from the mother, one of the genes, the other letter comes from dad, and so the two genes that are represented, or the two letters that are represented, are what we see in the offspring itself, and that's what we talk about as its genotype. So genotypes in conventional Mendelian genetics look like one of the three possibilities below. Either they're two capital letters, there's a capital plus a lowercase, or there's two lowercase letters. And each possible combination, we should know, has different terms that are associated. Homozygous, these are the genotypes that have the two of the same, right? Homo means same, so either two capitals or two lowercases. Homozygous organisms are also historically been called purebred organisms because they only show that trait and can only pass on that allele or that version of the trait itself. Heterozygous then, hetero means other, and so they're going to have a multitude or a different combination of those two letters. So one will be capital, one will be lowercase. And historically, these guys have been referred to as hybrids. They are a combination of the two traits, which means they can then pass on one or the other when they go to have offspring. So homozygous is two capitals, heterozygous is a capital and a lowercase. Homozygous can also mean two lowercase letters. All right, and these are the genotypes. These are the actual genes or the actual alleles that are present in the organism's genetic makeup within its DNA code. But then Mendel also comes in and says, all right, well, because each organism has two alleles for each trait, then how do we know which allele gets expressed? And that's where we talk about dominant versus recessive. So in order to show a dominant trait by traditional Mendelian genetics, that heterozygote organism will always show the dominant because it has one capital letter, because it has a dominant allele. So homozygous recessive, in order to show that recessive trait, you have to have both copies. All right, so dominant will always show through, will literally dominate or overexpress over the recessive trait, unless all that's present is the recessive itself. So again, one allele masks or hides the other. That's your dominant. The one that gets hidden is your recessive. And so we conventionally use capital versus lowercase letters to represent what's the dominant feature versus what's the recessive. So capital is the dominant, recessive is the lowercase. So your dominant gene is the one that overshadows the other. So let's take an example. Your Angus cattle, black is dominant. So that means genotypically that black cattle can either be homozygous dominant, so two big B's, or it can be heterozygous, the big B, little b. Red cattle, on the other hand, are recessive, and so these guys can only be two lowercase, so long as I mate two red cattle together, I will always get red cattle. But if I mate two black cattle together, that doesn't necessarily guarantee I can't get a red one if they are both heterozygous individuals. your recessive gene is the one that gets overshadowed. As a result, you need to have an organism have both lowercase letters or both recessive alleles in order to be showing the recessive trait. Okay, so example, black wool is recessive to white. All right, so a white sheep can either be homozygous dominant, so 2 capital W's, or can be heterozygous. But that black sheep, always recessive. Okay. So we're talking about genotypes, but we're also relating them to the phenotype. So we're talking about the coat color, the fur color, the wool color, and us, blue eyes, striped fruit, yellow flowers, doesn't matter. Whatever the organism looks like, that's its phenotype. But it's all based on what its alleles are in its genotype. Alright, so here's kind of that comparison. So when we look at this, your phenotype is the actual color it is. So purple flowers versus white. When we do phenotypic ratios, here's your traditional Mendelian genetics. Alright, 3:1 is the ratio, assuming we mated 2 heterozygous individuals together. But I can also talk about genotypic ratios. So here's my homozygous dominant, here's the 1, my 2 heterozygous, 2, and my homozygous 1. So my genotypic ratio is a 1 to 2 to 1 versus the phenotypic being 1 to 3 to 1. Alright, the last of Mendelian hypotheses comes with the idea of what happens when we go to make offspring. And this idea ties back to Morgan's gene linkage. If everything follows traditional Mendelian genetics, those 2 alleles for a particular character should segregate or should separate when we go to creating gametes, so that each gamete contains 1 allele for every character that next generation of organisms has to have. And assuming that this is normal genetics and not linked genes, then each of those alleles should have a 50-50 shot of being exactly like the parent or being some recombination of the 2 parents' genetic material. So what we're looking at with Mendel's 4th hypothesis coming from his pea plant experiments really are these 2 quintessential laws of genetics, and that's the law of segregation and the law of independent assortment. And these 2 laws really come back to meiosis, right? The 2-step cell division that gets us to our gametes, or our sex cells, your eggs and your sperm. And this is also going to be what allows us to use the Punnett squares we'll talk about in a few minutes to predict the outcome of genetic crosses. And really what these 2 laws say is that when I go through meiosis, my alleles, or my 2 letters for a particular trait that I got from my parents, are going to separate independently of each other to go ahead and make my sex cells, so that when I pass on my genetic material to the next generation, to my offspring, that they are only getting 1 copy or 1 allele for, say, your hair color trait. The other half is going to come from the other parent. So the law of segregation really is talking about the fact that you as an offspring have inherited 2 copies or 2 alleles for every gene, every trait that you have. One of those alleles came from your mom, the other allele came from your dad. We bring them together, and now they work with each other to give you the phenotype or the physical manifestation of your genes that we see. Okay? And law of segregation really gets demonstrated nicely when we talk about monohybrid crosses, which we'll talk about in a minute, where I'm looking at a single trait and I can use things like those Punnett squares to say, all right, well, if I know that these 2 alleles that this parent is going to pass on have to segregate when they go to make their gametes, then I can treat them as independent pieces that they can then pass on. And that's really kind of what your law of independent assortment is saying as well, is that when we go through and make those sex cells, that all of the alleles that I could possibly pass on for every trait that I can pass on to the next generation, they all are going to assort independently, and so I can treat them as independent individual units that can get shuffled around to make the offspring of the next generation. And your dihybrid crosses do a really nice job of sort of demonstrating that idea as well.